So FLT3, or I'm going to call it FLT3, are common mutations in AML. They activate a receptor tyrosine kinase, and for that reason, there have been drugs developed to inhibit the function of this tyrosine kinase. Those are important both diagnostically, prognostically, and therapeutically, because FLT3 really is only mutated in acute leukemias. FLT3 internal tandem duplications are both very common. About quarter of patients will have these mutations at diagnosis, and they portend a poor risk because they have a higher rate of relapses. And if patients relapse and have a FLT3 mutation present at the time of relapse, their outcomes are really quite poor with existing therapies. And that's one of the reasons that we've been interested in developing FLT3 inhibitors to improve outcomes in therapy. The big challenge at present has been that existing chemotherapy really doesn't work very well. It's highly toxic, and when patients respond to the regimens, and they can respond, they're generally not durable. So if anybody has a FLT3 mutated AML and relapses, we can give them chemotherapy and they can go into remission, but it usually lasts for such a short period of time that you can't take that response and then go to a bone marrow transplant, which might cure the patient. So the clinically meaningful responses to existing therapy are really quite rare. And these patients, once they have relapsed or refractory disease, their survival is really quite short. So one of the reasons we wanted to develop FLT3 inhibitors was to see if we could use targeted therapy to improve outcomes, both in terms of improving the likelihood of response and maybe better durability, increasing the likelihood of transplant, or for patients who aren't transplant candidates, to give them a reasonable option in terms of low toxicity therapy that still gives them the meaningful benefit in terms of clinical outcomes. So gilteritinib is a selective and it's a potent inhibitor of FLT3. The selectivity is important because drugs that are inhibitors of kinases but not specific for one or really selective for one target tend to have a lot of off-target effects, and so side effects can be limiting and may keep you from being able to give enough of the drug to inhibit the target very potently, and that's really where you're going to see anti-leukemic effect. So the advantage of gilteritinib is you can see what a FLT3 inhibitor can do to slow down leukemia or even put it into remission. And the second time, the advantage is that it's been designed to be active against all the different kinds of FLT3 mutations we see clinically. And there are various different kinds of mutations that we run into. The FLT3 ITD is the one we see most commonly, but there are activating mutations in the tyrosine kinase domain that are not so important prognostically, but they're important clinically, because if we use FLT3 inhibitors, some of them will actually select for mutations in the tyrosine kinase domain that confer resistance to those drugs. So this drug is active both against FLT3 ITD and against FLT3 TKDs, and for that reason we don't tend to see the resistance by that mechanism, and that may lead to either more likely responses or more durable responses. So the ADMIRAL trial is a pivotal study of gilteritinib. It's a phase 3 study in relapsed and refractory FLT3 mutated AML. So patients, when they enrolled to the study, had to prove that they had a FLT3 mutation at the time of resistance, not just that it was there at the time of diagnosis. And we randomized 371 patients to either gilteritinib as a single agent, so it's a once-a-day oral tyrosine kinase inhibitor, or the investigator's choice of what they thought was the most appropriate standard chemotherapy from a list of 4 different treatment choices, 2 of which were intensive chemotherapy and 2 of which were low-intensity chemotherapy. So these included everything from FLAG-IDA or MEC, which are very intensive regimens, to low-dose RSC or azacitidine, which are lower intensity. Patients were randomized 2-to-1 to either the FLT3 inhibitor or the chemotherapy, and there was no crossover from one arm to the other. If patients responded to gilteritinib, they could actually go on to transplant and go back on the drug after transplant. And the primary endpoint of the study, there were actually 2 primary endpoints, both a response primary endpoint in the gilteritinib arm only, and that was really just used for regulatory submission of the drug. But the real primary endpoint of the study, the co-primary endpoint, was overall survival. And the study did not change conduct even though it was positive for both primary endpoints. So it enrolled to its completed sample size, and the final analysis was not triggered until 258 survival events had occurred. And at that point we looked at the overall survival, and we're very gratified to see that gilteritinib showed improved, statistically significantly improved overall survival compared to chemotherapy, and that was true regardless of the intensity of the chemotherapy chosen and a number of other metrics. So the survival in the chemotherapy arm was 5.6 months, and in the gilteritinib arm was 9.3 months. So we not only saw more than double the remission rate on the gilteritinib arm, but also better survival and higher transplant rates. And again, that's important because that's the only chance of cure in this disease. And then one frustrating thing about this is, despite these improved outcomes, we still are finding that survival in this population, even with advances like gilteritinib, is still relatively poor. So we really need to take effective therapies like gilteritinib and move them into the frontline study. And that's really where we're headed next in terms of our future approaches. The last thing to comment on is just gilteritinib did this with actually less toxicity than existing chemotherapy. So if we look at both exposure corrected toxicity for either severe or grade 3 or higher or SAEs, they were lower in the gilteritinib arm than in the standard chemotherapy arm. The common toxicities we did see were cytopenias, LFT abnormalities, but really we saw relatively less in the way of serious infections or serious bleeding and other complications of existing chemotherapy. So I'm actually presenting data at this meeting also looking at that very question, combining the same dose of gilteritinib that we use in the relapsed refractory setting as a single agent with intensive frontline chemotherapy, cytarabine and daunorubicin, or idarubicin in 7 and 3. And what we found is that that's fully feasible at, again, full doses of all of the drugs. And the remission rate looks extremely good. 93% of patients enter a complete remission with this approach on the study in the patients who have FLT3 mutation. So it's a little bit lower if you look at all patients, but that sort of makes sense if you think about it, because gilteritinib really, its activity is limited to patients who have FLT3 mutation in the relapse setting or the refractory setting. So it's not surprising that we would see a very high remission rate in patients with FLT3 mutation, chemotherapy and gilteritinib, and just kind of comparable numbers to what we would see in chemotherapy only.